Hello, ASPET members, and welcome. My name is Yolaine John, and I'm the meetings coordinator at ASPET. Thank you for joining us for the Focus of Pharmacology series, where today our focus is on the coronavirus. We want to thank our sponsor, the Burroughs Wellcome Fund. You can learn more about their Innovation in Regulatory Science Award at the link here. The deadline to apply is February 12th. Before we start, just a few housekeeping items. You will note that all attendees are on listen-only mode. Some of you have already found the chat function where you can send messages to other attendees. Be sure to select the correct recipient option above where you type your message. There is an option for chat with panelists and chat with attendees. If you have a question for the presenter, please use the Q&A function, which can be found at the bottom of the Zoom window on a desktop or at the top of a tablet. You can submit questions throughout the presentation. While in the Q&A area, you can also vote for questions you most want answered by clicking on the thumbs up icon. The little number tells you how many other people also want this answered. By attending this session today, you can send to ASPET's Code of Conduct and having your questions and chat recorded. A recording of today's session will be made available to ASPET members next week in the ASPET Connect library in the Focus of Pharmacology community, which is also where you can find the recordings from our past sessions. While in today's session, it is being recorded, to maintain the scientific integrity, we ask that you refrain from grabbing screenshots or video capture of any of the scientific presentation without the speaker's permission. To help you differentiate what you do and don't have permission to capture, look for one of these two icons on the speaker's slide. A camera with an X means do not record in any way. A camera with a check mark means you can screenshot it, but sharing and remixing are only permitted with appropriate attribution. In the absence of a visible icon, all forms of capturing the slide are prohibited. At the conclusion of the webinar, we welcome everyone to continue the discussion in ASPET Connect. Also, you'll receive a survey link from us. It should take about 2 minutes to complete. We look forward to hearing your feedback. All right, let's begin. I'd like to introduce our session moderators. We have Dr. Michael Jarvis and Dr. Cheng MH. Dr. Jarvis, let me put it over to you now.
Hello, everyone. My name is Mike Jarvis. I'm a member of the SPET Program Committee, and it's my pleasure to introduce Dr. Yvette Bahar today as one of our speakers in our COVID-19 series. Dr. Bahar is the Distinguished Professor and John K. Veery's Chair and Founding Chair of the Department of Computational Systems Biology at the University of Pittsburgh. In addition to her affiliation at Pitt, Dr. Bahar holds director or principal investigator roles in many different computational biology research organizations across the planet, including 2 NIH-funded centers at NIGMS and NIDA, as well as she's a founding committee member of the Carnegie Mellon University and University of Pittsburgh PhD program in computational biology. I think most folks are familiar with at least some of Dr. Bahar's groundbreaking work in the area of computational biology. And I believe people are also aware that she was inducted into the National Academy of Science in 2020. So without further ado, I'll turn the presentation over to Dr. Bahar, who's going to talk to us about the superantigenic region of SARS-CoV-2 spike and its association or mechanistic underpinnings with multisystem inflammatory syndrome and a similarity to toxic shock syndrome. So over to you, Dr. Bahar. Thank you so much.
Thank you. Thank you. Okay, I think you are able to see my slides now. So today I'll talk about a superantigenic region in SARS-CoV-2 spike and how we can develop new therapies taking advantage of the new knowledge of this superantigenic region. So mainly I'm going to summarize the lessons we learned from our computations and experiments. And you can see here a very long list of collaborators, because I'm going to talk about various studies in which we have co-authored with those individuals from the University of Pittsburgh, where I am, from Cedars-Sinai in Medical Center in LA, and Wittenberg University, Martin Luther University in Germany, and also Stony Brook in Long Island, in New York. So we have essentially, I would like to briefly review a few major features that we have learned. And then I'm going to proceed to the new findings and how we can, a new antibody that we discovered, which I hope you will be interested in. So essentially, SARS-CoV-2 spike interacts with many proteins of the host cell, including proteases, T-cell receptors, and monoclonal antibodies. The way we started to this problem to start investigating the spike behavior in general was when we learned about this multisystem inflammatory syndrome in children, shortly called MIS-C. We were intrigued really, and we wanted to see what is the origin of that type of behavior. And later on, that type of syndrome in children was also found to be taking place in adults with severe COVID-19. So that was the original study we published actually in PNAS, where we, we wanted to bring to the attention of the community the existence of a insertion in SARS-CoV-2 spike that exhibits superantigenic character. So I'm going to talk on that first. And this is like the acknowledgement slide for me. I'm showing here 2 students who were involved. I mean, a student and a research assistant professor in my lab, Mary, who were involved in this study. And this was also in collaboration with the lab of Moshe Arditi and Rebecca Porritt in his lab at UCLA. So as background, I'll give you a brief outline of what MIS-C is about. And many of you may already know, but just brief recap of adaptive immune response, what superantigens are, how they are different from antigens, and also a little bit more of background on the spike sequence and structure. And again, it's not anymore only children, it is also observed in adults, and this is called MIS-A. Now, this has been newly recognized, a new condition in which the children originally and now adults, they present a constellation of symptoms, including hypotension, hyperinflammation, and these symptoms that are observed in those patients, they strongly resemble what is called toxic shock syndrome, TSS, which is usually triggered by what is called superantigens. So here are the symptoms for those of you who are interested in the, the type of manifestations: high fever, skin rash, etc. And originally, this, you can see here how MIS-C and the pediatric TSS, toxic shock syndrome, show very similar characteristics. And we wanted, based on that, we wanted to understand what is happening. And for those of you who are also interested in an overview of the topic, there is a recent review which talks about MIS-C in children and adolescents. And you can, it is not only, it is a systemic response, actually. That is where the problem lies, because it is not only at the original insertion of the virus, but now the pyroptotic cell death due to high inflammation, the discharge of DAMPs, damage-associated molecular patterns, molecules. And then there is a cascade, actually, that is being perpetuated and enhanced by antibodies. So we would like to understand the origin, how it starts at least, because we would like to stop it, right? Now, again, a brief review of cytotoxic adaptive immune response. I'm sure many of you are familiar already. What happens is that we have the MHC molecules, major histocompatibility complex, which present the peptide here shown in red to the TCR, T-cell receptors attached to the T-cell. So there is a cell-cell communication for whenever there is an antigen. So this is the peptide. And we know that in the TCR, in the variable domains of the alpha and beta chains, there are those complementary to determining regions that specifically recognize different peptides. So this is the classic cytotoxic adaptive immune system response in response to antigens. Now we're talking about superantigens. Superantigens are not peptides anymore, but these are bacterial proteins or viral proteins. So we are talking about proteins. And actually the bacterial superantigens have been very widely studied. Examples are the toxic shock syndrome toxin B, staphylococcal enterotoxin B, etc. We know much less on viral superantigens, but there are also a few of them, as I'm going to describe. And what happens is that in the presence of such proteins, you have a very strong response, immune response, that could be excessive and lethal, leading to the toxic shock syndrome. So it is, in this case, we know that, for example, in the bacterial toxin, we have a huge expression of T-cell receptors, V beta domains, from 7% to 50% in the cellular environment. And this also shows some symptoms similar to Kawasaki disease, but it is different. Now we are interested in viral superantigens. So what viral superantigens do? They bind either TCRs or MHC2 molecules, and most commonly TCRs and their variable domains of the beta or alpha chains, one or both of them. So in some cases, they also bind MHC molecules, MHC class 2, but the binding takes place outside the conventional, peptide binding groove. So it's different. Here you can see a few structures that have been resolved. And in particular, I would like to draw your attention to SEB, staphylococcal enterotoxin B, which will be of interest, as you will see. So how SEB interacts with TCR beta chain here, or SEH is another subtype, which is shown here to interact with both alpha and beta chains. And here are the epitopes that you can see. So essentially, we know that the superantigens have a very high tendency to attract TCRs, T-cell receptors, and that leads to escalation of immune response. So the question that we posed were, does SARS-CoV-2 spike, does it possess any superantigenic features, since the symptoms that we are observing in those patients are very similar to those that would be elicited by superantigens. So does the spike interact with the TCRs? And can we identify any segment sequence similar to known superantigens? And also there is, SARS-CoV originally in 2003 has been thoroughly examined. And indeed, we are taking advantage of the knowledge that accumulated then. So we're going to see that there were superantigenic regions characterized then, and we will compare with SARS-CoV-2 spike. And also we will try to understand the effect of variants. These days everybody is talking about variants, so it's a really hot topic. Now, again, very, very quick overview of the SARS-CoV-2 spike. You can see here this two spikes actually that bind the human receptor ACE2. ACE2 itself is a dimer, and it has two helices embedded in the membrane, supported by a transporter. So that is the picture we usually know. And as I mentioned, it interacts not only with ACE2, but also proteases, T-cell receptors, antibodies, and other molecules like neuropillins. Now, of course, this is the big picture of the virus. And what we are doing is essentially focusing on the spike interacting with the human ACE2, as you can see here. Now, a closer look at the spike shows that it is a trimer. Now, each monomer is a different color here. And we have I colored here in green the parts that are interacting with the spike, the RBD, receptor binding domains of the three monomers. And this region undergoes a conformational change from a close to an up state in order to expose a residue, K417 and others, to enable a tight interaction with ACE2. And in this position. So what I would like to emphasize here is that each monomer is composed of two subunits, S1 and S2. And in the middle, you have the cleavage site between the two subunits. RBD, the receptor binding domain, is part of S1. And you can see where, roughly speaking, this is the region where S1 is located. And this is the region where S2 is located. And there is a cleavage here at a position almost here, as you can see from my mouse, which separates the two trimers after recognition by ACE2 and proteolytic cleavage by host cell proteases. Now, here is another perspective, a very pretty figure from Scientific American, ACE2 and spike interacting. The protease, as I mentioned, is critically important. It binds the cleavage site to separate the spike into two parts. Now, where does the protease bind? The protease, if you compare the sequence of SARS-CoV-2 to other SARS family viruses, spike, you can see that there is an important difference. There is a segment, PRRA here, that is inserted in SARS-CoV-2, which is absent in all other SARS family members. That's the unique features of this current SARS-CoV-2 spike. And then right adjacent to this PRRA insert, we have the RS cleavage site. That's where the subunit S1 and S2 separate. So this is the insert that I mentioned, and we will come to that because it is very important. So if you compare SARS, the 2003 and current SARS-CoV-2 spike glycoproteins, there is a significant structural difference at the region of this insert. So that is something to pay attention. Again, as I mentioned, when we have the protease cleavage, this is where the PRRA insert is, and just next to it is the cleavage site. So the trimer in here, I color, spectrally colored one of the monomers, starting from the anti-D blue to the C-terminus red, and the other monomers are white and gray. After separation, you see what this is the subunit S1 that separates after cleavage. Or if you wish, you can look at the trimer cross-sectional area. If we cut from here, you can see that those blue PRRA segments are quite exposed in this cross-sectional view. You can see from here. So that is, and then this is the other part. This is the S2 trimer now, which forms a elongated shape and undergoes a conformational change to serve as the fusion machine, as you know. Okay, so it was, when we got interested in MESSE and this possibility of toxic syndrome, we also noticed at that time a paper by Jean-Pierre Changeux in compte rendu, where he, in the context of another hypothesis, he brought attention to the fact that the SARS-CoV-2 spike sequence near this PRRA segment had some similarities to known toxins. And so here you can see a bigger representation, and we added a few more actually, including an HIV glycoprotein that also contains a known superantigenic motif. So we can see similarities between our sequence in this small stretch of 10 residues, approximately, and other toxins that or superantigenic fragments. So we wanted to look more carefully, and then when we examined in a more systematic way, what we found that the SEB that I mentioned, remember I mentioned the staphylococcal enterotoxin B, the segment from of SEB from 150 to 161 shows a very high similarity to a segment of the SARS-CoV-2, and it is definitely very different from the CoV-1. And what is even interesting is, Mary in my lab, she came up with this idea of inverting the sequence, like palindromic sequences. And you could see that actually there is even another segment of the SEB far away. It is connected here, but in the case of SARS-CoV-2, there is an insertion, but that is also similar. And what was even more striking is that not only the sequence, but the two structures, that of the superantigen SEB and this motif from residue 661 to 685, they showed very similar three-dimensional organization. So we have now we discovered this segment in SARS-CoV-2 spike, which shows both sequential and structural similarity to this bacterial superantigen SEB that is known to cause toxic shock. So to us, we say this ought to be a superantigen segment as well. And we wanted to see whether it has an ability to bind TCRs like other superantigens. And we could see that, all those basic arginines are basic, amino acid, and including the previous serine, they are closely involved in a tight interaction with the V beta chain, which is typical of superantigens. Then we went back to what's known in the literature for SARS-CoV of 2003. And in this study in 2004, actually, they did analyze the sequence to find whether there are toxin-like segments. And indeed, in all proteins, actually, of the virus, we focus on the spike. And these are the segments that are superantigenic. And in particular, two motifs shown in green here caught our attention because they are intracellular adhesion molecule, one ICAM-like segments, which could serve as adhesives, if you like, for adhesion and stabilize the complex. Now, when we looked at their positions on the counterpart in our in SARS-CoV-2, some of them had low sequence identity, but one of the ICAM, this intracellular cohesive adhesion molecule segment had relatively high sequence identity, and it is located just next to the PRRA region, superantigenic region. And when we examined, closely whether this ICAM1-like region, shown in orange here, one part of it, also interacts with the V alpha chain of the TCR. So not only does the PRRA show tight interactions here, but we also have an additional segment which acts as a glue for further stabilizing this interaction. And there are more details, but I would like to just mention the major PRRA segments complemented by this ICAM1-like segment. Okay, and then our collaborators in Germany actually, they examined this T-cell receptor repertoire of COVID-19 patients. And they found as a typical response of superantigens is to observe a skewing in the T-cell receptor expression levels. And they found that particularly there were a few, these are the gene notation for the T-cell receptor variable domain beta chain 5-6 subtype, and others that were very strongly expressed in the case of severe or hyperinflammatory patients, shown in orange, as opposed to moderately or mildly diseased patients, showing that there is a distinction between, in severe case, we do observe this type of T-cell receptor skewing in support of the superantigenic effect. This is already published, this part about 6 months ago. So for more details, you can go there. Now, now that we have discovered such a superantigenic region, the next question is, can we how can we exploit that to perhaps design antibodies that would be targeting this superantigen-like region to preempt a possible superantigenic reaction, right? And we also note that the superantigenic region includes this PRRA segment. Remember that this was next to the cleavage site. So the question that we asked was, is it possible that an antibody could have a dual effect, not only neutralizing the superantigen, but also preventing protease binding? So then we remembered that Bettina Fries, who is also our collaborator at Stony Brook, she's an expert on bacterial superantigen SEB, staphylococcal enterotoxin B. And she has identified 3 antibodies here shown in different colors, blue, pink, and blue, I'm sorry, SEB is in blue or here in gray, and the antibodies are in yellow, green, and magenta, which bind different parts of SEB. So here are the 2 color-coded antibodies published 5 years ago. And then we said, okay, since those antibodies, they neutralize SEB, and we have a very similar segment on our spike, is it possible that one of them could be neutralizing the spike as well at this position? And among those 3 SEBs, 3 antibodies, which we show here again, they all bind different regions on the bacterial toxin. But one of them, here in blue, is the motif that is similar to our superantigen on the SARS-CoV-2 spike. So 63 among the 3 antibodies is the one that binds exactly that SAG motif that is similar to our superantigenic fragment. And here is a large view. You can see what happens is that here the SEB has also a bunch of basic residues, lots of lysines. In our case, it's arginines, here it's lysines. And then it's on the other side, on the antibody, you can see many negatively charged acidic residues. So it's a very tight binding there. And we know that our superantigenic region has also many positively charged, a polybasic segment. And of course, when we tried in modeling and simulations to see whether it binds here, as expected, we could see that there is a very strong interaction. You look at the type of interaction, how this thing really closely interacts with the SARS-CoV-2 spike region here, and the light and heavy chains of the antibody 63. And here you can see more details. This part is the spike, and now here you can see the counterparts in the antibody. More importantly, by the way, these more details can be found in this bioRxiv report that we uploaded a month ago approximately, and then we have a manuscript under review. More importantly is the experimental confirmation, actually. So at UCLA, at Cedars-Sinai and UCLA, they performed this type of dose-dependent experiments with increasing concentration of 63. This is the control, and this is in the presence of 63. And you can see that gradually the infection percentage decreases. And these are measured based on two different measurements. One is the spike, and the other is DS, double-stranded RNA measurements. In here, I'm not sure if you can see clearly, and I'm sorry for the quality, but what, and in the paper you can see more details. In the presence, if you have a very small amount of the 63, the red dots that you see here are the double-strand RNA that is in the cell. And this gradually decreases, and you have very few left with increasing concentration of 63. And same thing for the spike. Here you have lots of red points. And as in the isotope control, it's about the same. Of course, for the isotope control, you have no change, but here in the presence of 63 increasing concentrations, the red dots gradually decrease. So these are experiments conducted in Vero A6 cells for those of you who are interested. And here's a brief summary of the way those fluorescence images were taken. So each image actually, maps up to a single point, and this is the most probably meaningful result. We have an inhibition of the infection by about 80%. So now this is one of my first conclusions. I'm going to give you the conclusions like that. So 63 is a SEB monoclonal antibody significantly reduces SARS-CoV-2 infectivity. This is predicted by computations and confirmed by experiments with live cells infected by virus. Now, then a question that arose actually, we know that spike is a glycoprotein. It's glycosylated. It has lots of glycans. And actually there is one here next to N603, which is spatially neighboring our superantigenic region. And the question was, does the existence of this glycan possibly prevent 63 binding? And when we did our modeling simulations, we actually observed that no, SAG is still accessible, and the presence of this the glycosylation site here is even good for binding. It even further stabilizes. It binds, forms hydrophobic contacts. So we don't anticipate any, blocking effect of the glycans here. On the contrary, it may even further help. Now, the next question we asked was, what is the mechanism of action, right? Yes, we know that this is the superantigenic region, but the type of experiments we did were to measure viral entry. And what we are observing is a reduction in viral entry. And remember, the viral entry needs the separation, the cleavage of the subunit by the protease, and the cleavage takes place at this site that overlaps with our superantigenic region. Okay, so is it that we have 63 interfering with the S1S2 proteolytic cleavage site and preventing the protease activity? Is it the reason for observing the reduction in viral entry? So we wanted to examine that more closely. So here is just a background slide showing that, you may have two different types of entry of the viruses. As you know, it could be through fusion, the way I described, and you require here again this proteases cleavage, or it could be through endocytosis as shown here, but you still need those the cleavages. So this PRA site, or its first neighbor RS, where the cleavage takes place, is critically important for infection, for the discharge of the RNA of the virus to the cell interior, double-stranded RNA. All right. So and then we started to examine more closely where the proteases bind now. And we consider two proteases. One is TMPRSS2, and the other one is furin. And they both bind, actually, in our models, we saw they both bind very tightly. And they make lots of hydrogen bonds, salt bridges, etc. And furin even binds more tightly than TMPRSS2. Here you can see also a view how this polybasic segment, the PRRA, closely here is the spike, its PRRA segment, how it interacts with furin. And indeed, this type of segment, polybasic segment, is called a furin cleavage site, because furin itself also has a bunch of negatively charged amino acids that exactly position themselves, and this is a very strong attraction. Now, when we, so there is, the question is 63 binding strongly enough to compete with furin in particular, and even TMPRSS2. So here are the results, actually, from binding energy calculations. Yes, furin binds stronger than TMPRSS2, but the 63 is comparable. So it is gonna effectively, there may not be 100% prevention, but there is a competitive binding, which is gonna reduce the way we saw already in experiments. So we conclude that 63 does compete with protease, and it is probably the mechanism underlying the reduction in infection that we have observed. So then one might ask, we already have lots of monoclonal antibodies, right? That have been already discovered, structurally resolved. And here we show actually the superposition of many of them here. One of them is ACE2, not the antibody. This is where these are all the regions of the spike protein that bind, for example, ACE2 or a bunch of antibodies. Okay, one of them, for A8, actually binds the NTD, N-terminal domain, and all others bind the RBD. So, do we need one more? You might ask. We already have many. And the answer is yes. Why? First of all, in all those studies, all the structures that I've shown here, if we go to the original studies, they have been resolved by cryo-EM. And in all cases, the PRRA region, this superantigenic region, was mutated. So that is, if there is any effect originating from this unique segment, superantigenic segment, it is mute here. You can't observe because it has been mutated. That's one thing. And they all bind and these are the corresponding references for those of you who are interested. And furthermore, they more or less, except for A8, one of them, this one, which binds the N-terminal domain, they all more or less bind the same sites. And this is a problem because you see if K417, for example, or many others here that we observe, have been observed in mutants, variants, and in the presence of their substitutions, the question arises whether those variants can then evade those antibodies. So this brings us to this very important topic, the emergence of SARS-CoV-2 variants. As you can see in this image, you can see the spike protein, and where in the spike we have had important mutations. For example, in this RBD, receptor binding domain, as you may have heard in the news and in the literature, this is a very important mutation, and the 501 residue asparagine being replaced by tyrosine, which is in the UK variant as well as the South African variant. That one, replacing at the interface an asparagine by a tyrosine definitely increases the binding affinity, and hence all the concerns about the higher transmissibility or infectivity of the variant. And another one is 484, which is in the South African. And 2 days ago, they've also said that the UK variant has now also developed one more mutation, which is exactly that site. That is also critically important because it is, again, at the RBD, and it is a binding site for many antibodies. Now, replacing a negative charge by a positive charge, you would expect it to be consequential. Or another one, the early discovered, 614G. That one, originally, many people were worried about that one. But it turns out this is not so consequential, really. It is not involved in the interaction with the ACE2 or antibodies, but it might affect the flexibility of the spike. Okay, so based on that, we have actually recently examined what happens with the South African variant in an article that we deposited, also uploaded to bioRxiv for those of you who are interested. And I'm just going to give you one result from there. We examined all those different antibodies, including the Regeneron cocktail. It has two antibodies, 10399 and this 10987, that bind different sites, more or less. And there are many others shown from different perspectives. I'm just going to give you the final result. Of the 11 examined cases, 5 were not that strongly affected. 2 may even be better binding, so they will be effective still with the variant. But 4 of them showed a decrease in binding affinity. Among them is one of the Regeneron antibodies. And what happens is, in this case, the Regeneron is a cocktail, right, based on 2 antibodies. The other one is fine. The other one was not affected. So the cocktail is still effective, but that is a red flag. We would like to understand better what is happening. And when all those antibodies are all targeting the same site, more or less, RBD, which is very fast mutating, that is probably not a good strategy. Instead, we may want to have another monoclonal antibody, like R63, that binds a completely different region, so that the probability of evading the antibodies, the probability of the variant to evade them, will be lowered, if not completely removed. So that completes my presentation. I was asked to give a talk of 40 minutes or 45. So here is a summary. We have, as I showed earlier, the spike is a toxin-like superantigenic site. This site binds TCRs, T-cell receptors, to possibly elicit hyperinflammatory response in MIS-C and MIS-A. And it also binds the antibacterial monoclonal antibody 63, which competes with the proteases to inhibit viral entry. So in this figure description, you see we are talking now of 3 different cells coming into, this is the virus, and then T cell and host cell coming into close proximity. You have ACE2 binding the spike, and the protease is in purple here. After cleavage, it is broken into 2 parts, S2 trimer and S1 trimer. And this S2 trimer, the pre-fusion conformation, undergoes a conformational change to this elongated shape, and which enables the fusion of the viral membrane and host cell membrane. And in red are all those superantigenic regions, whether they are in the intact spike or the cleaved one, S1 trimer, they are going to attract the TC receptors, which may then escalate into a superantigenic response. But knowing that this superantigenic site has same sequence as a bacterial toxin, an antibody that works with the bacterial toxin also binds the same site here. And it also competes with protease binding, which is probably the reason for the observed effect, a reduction in infectivity. And so 63 right now, further experiments are in progress to see how 63 may be used in cocktails as well, and to identify exactly that this is the binding site. So this is my lab these days. This is the way we meet. I would like to thank you all for your attention, and I had the pleasure and honor of interacting with many people, starting with Mary Cheng, an extremely talented researcher in my lab, and James Krieger, who also completed his postdoctoral studies, and many students and research associates and collaborators. Moshe Arditi was the person who brought the first problem to our attention, and as an infectious diseases expert, also an immunologist, he has been really extremely helpful. We learned a lot, and we had also the pleasure of interacting with many other collaborators in this present study that I presented. Thank you very much for your attention. So I'm happy to answer questions.
Dr. Ma, I think you're.
Yeah.
Oh, thank you so much, Dr. Bahar. This is a really wonderful and beautiful presentation. It's kind of open eye presentation that I know the concept of computational biology for quite some time. I also participated in some of the studies, and this is the first time I learned how to integrate that into the pharmacology. And thank you so much. And now I open the question and the answer session. I always try to take advantage to ask you a first question. What you did give us such wonderful wonderful lecture regarding the combination of integration of pharmacology and computational biology. What my question would be, what you see the current challenge in the computational biology, and how to integrate that into the pharmacology? It's a broad question. I know it's a broad question.
Yeah.
Go ahead.
So it is a very broad question indeed. But I would like to say that, computational biology itself is also very broad. What we are doing in general is I divide into two groups: molecular pharmacology and quantitative systems pharmacology. So molecular is, everything you've seen here is more molecular modeling and simulations. But in many cases, that's not enough. We also have this second group of studies, which we call quantitative systems pharmacology. These are more data-driven. So, we use machine learning algorithms to learn from existing databases. And so that is the basis of, discovering repurposable drugs, identifying targets, or understanding mechanisms. So it is quite, computational biology in itself. There are those who are more experienced in the biochemical, biophysical aspects. Others are more into genomics or image processing analysis. It is in itself very broad. And like in all other approaches, it helps a lot to use multiple tools that we have, combine them. So here, this is also, what I presented is at the intersection of computational biology, but also there is a lot of immunology and other challenges for us. We make lots of predictions, and we would like them to be tested, validated. So we are extremely grateful to our collaborators in Germany, in UCLA, who have been testing all those predictions. We could, then everything becomes meaningful.
I see. Thank you. Now, let me get into the question I collect through the question and the answer session. One of the audience asked, do you have any experimental evidence that supports that N-glycosylation near to the antibody binding sites does not decrease or increase the antibody binding to the spike protein, such as using enzymatic deglycosylation and checking the antibody binding? It's a long question.
That's a great question. We didn't do this type of analysis. We were more interested in the particular region, that we identified to be a target site. This region that contains the insertion PRRA, and we also looked at the structures that have been resolved, and we know that we have already the position of the glycosylation sites of this glycoprotein. We already know that, and we wanted to see, compare with experiments to see whether there is an overlap, whether the region which we are targeting would be blocked by the glycans. So based on the cryo-EM structure of the glycosylated spike, we are good, but we haven't done any other experiments to really test the effect. This is a very good question, which probably needs further validation, but we had at least the cryo-EM structure to see that.
I see. Thank you. That's the question I have online. So I do have a few questions. So I want to ask you about. Sorry, I just being the devil's advocate here. So,
no, that's great.
I have a couple questions. I just need some clarification from you. For example, when you talk about the PRRA insert,
yes.
What's the just from the literature, what's the function of that PRRA?
Yeah, that's a very, actually, I had a few more slides, which I haven't shown.
Oh, okay.
Because I was concerned by the timing, but I can explain. So as I mentioned, that's, I'm glad I could clarify that.
Okay.
As I mentioned, this PRRA is unique to SARS-CoV-2. So among all SARS subfamily of coronaviruses, beta coronaviruses, only the current virus has this PRRA insert. But there are other beta coronaviruses, including human cold, coronavirus, MERS, many others. Interestingly, they do have actually some of them, like in the bioRxiv paper, you could see we have elaborated on that. Some of them have actually this a segment, polybasic segment at the same position. Exactly. Overall sequence identity is very low compared to SARS-CoV-2 glycoprotein, but at that region, at that position, structurally, they are very similar, and at that position, they have polybase. And what is that segment? Such a polybasic segment is actually a signature motif for furin binding. So it is even known in the literature as furin cleavage, site. So it is the signature of furin binding and cleavage site that is typical of many other beta coronaviruses, but not the SARS subfamily of coronaviruses. So it is, and since SARS-CoV-2 has this particular site, I think we need to take advantage of it. And what we have shown in also our bioRxiv paper, which you can find easily, is that 6D3 actually binds another human common cold viruses, which also have a similar polybasic insert at the same position. So 6D3 can have also the advantage of possibly neutralizing other spikes that also have such a polybasic segment. And it has, what happens is in 63, there is CDR2 actually, the complementary determining region 2, which is polyacidic. And this is a perfect match there. In the same way as furin has a polyacidic segment that binds the polybasic spike.
I see. Thank you. Here is the other question. With all of the mutations thus far, for entry, would it be more cost effective to develop polyclonal antibody instead of a monoclonal
Oh, yeah.
antibody.
Polyclonal antibodies is what we have in the sera, right? In what the immune system builds once we get sick. And naturally, we have this type of polyclonal antibodies. Monoclonal are used as drugs. It's another type of drug after all. But instead of a single type of antibody, to have at least two monoclonal, this is what is being synthesized. So it is not produced in response to the infection, but we are using as a treatment. So it is better to have a polyclonal antibody also, obviously. But if we have a really very specific one or two or even three of them in our cocktail treatment, I think it's gonna be effectively neutralizing. And indeed, that is also already seen, this Regeneron cocktail and the Eli Lilly, which Eli Lilly originally started with a single antibody, but now they are developing it into a cocktail because they have observed that it has diminished effectiveness with the variants. So it is obvious that you need more than one antibody if you would like to preempt the effect of current and future mutations.
Thank you.
Dr. Bahar, I have a somewhat naive question, but could you comment on the magnitude of the decrease in affinity for RBD that would be clinically meaningful, I guess is what I'm trying to get my head around.
So, that is, in this report, again, we submitted for publication and uploaded in BioCap. We gave numbers on the change in, binding affinity in kcal per mole. These are all models. I have to, please remember, and until these are experimentally verified, these are just giving guidance as to which antibodies should be really more carefully considered vis-a-vis the variants. Now, in our case, the change in binding affinity was really small. When we say 5 of them are weakened, this is not in terms of kcal per mole, it was like 1-2 kcal per mole, which is really small. And that could be offset by increase in their concentration, actually. So if you increase the dosage by a factor of 3, for example, you may alleviate that, this type of loss of function. You can have still effective antibodies, according to our calculations. So we are also in touch with another lab at Weizmann Institute, where they are directly measuring the binding affinities right now.
Thank you.
Great, thank you. Here is the other question. Will activation of furin can be used therapeutically?
So, the thing is, you don't want to stop furin usually, or the other protease, because they are required for other activities. So we don't want to do that, but we would like to block this region of the spike. So that has been the strategy.
Let's give another minute or two. We are almost at the top of the time. I have one quick question. You start the presentation about the hyperinflammation status, and then later on, I'm not sure that the hyperinflammation status also symptomatically relate to the severity of the disease.
That is what is being observed, when there is this type of escalation of the adaptive, cytotoxic adaptive response. Our collaborators in Germany, they did experiments with patients, and they have observed, I showed the data there, and it is in the PNAS paper. They have observed indeed a overexpression of specific types of TCRs, V beta chains, what they call a skewing in the repertoire of TCRs, which is a typical response of superantigens. But this was observed only in cases with severe COVID-19,
I see.
not in the mild or moderate cases.
Okay.
So there is a difference, you know, in the way this disease develops. In some people, there will be this strong escalating response, and in this case, it looks like there is exactly the same type of symptoms that would be otherwise caused by a superantigen. So that is
right. That makes sense. I understand.
Yeah.
It's the observation study always have some limitations, but that's what we have observed. Yes. Thank you. Okay. So we are at the top of the time, and thank you. I closed out of the question and answer section now. Thank you.
Okay. Thank you very much. Have a nice day.
Yeah. Melissa or Yolan?
Dr. Jarvis?
Dr. Bahar, thank you so much for very interesting
I hope it wasn't
presentation.
too computational.
No. I think it's quite valuable, and as you can see from the questions that were asked, I think it's extremely interesting. Thank you so much.
Thanks a lot.
Actually,
so
it's very interesting.
You will let me know how I can view the video, right?
We will send you a link so that you can
But it won't be accessible to the general community.
To ASPIT members.
Only ASPIT members, yeah. Okay. Sounds very good. Thank you very much. Have a nice afternoon.
Well, thank you everyone.
Thank you so much.
Bye-bye.
Thank you everyone for joining us today. Please be sure to complete our survey, which will open up automatically once you close out of Zoom. You can continue the conversation to this discussion in ASPIT Connect in the Focus on Pharmacology community. A recording of today's presentation will be loaded to ASPIT Connect next week. Be sure to tell all your colleagues. On February 12th, please join us for part 5 of our trainee career development series, How to Use Social Media and Networking for Your Job Search. You can register for this session in ASPIT Connect. I want to thanks again to our session moderators, Dr. Ma and Dr. Jarvis, and our speaker, Dr. Bahar. And thank you to our Burroughs Welcome Fund, our sponsor. Thank you everyone for joining and have a great day.
Thank you.